Hello, my name is Adèle Meroux and I'm a second-year PhD student. And I would like to thank the organizing committee for allowing me to present my work today that deals with the angiogenic properties of papillary and reticular fibroblasts in vitro. I'm carrying out this work under the supervision of Laurent Muller at the Collège de France, of Florence Ruggiero at the Institute of Functional Genomics at Lyon, and of Brigitte Kloss and Cyril Borde at SciLab at Brive-la-Gaillarde. So what is well known about the dermis? It's that it can be subdivided in two main compartments, a superficial one called the papillary dermis, separating from a deeper one called the reticular dermis by a vascular barrier. It is well known that they show different extracellular matrix or organization and composition, and it's also well known that they have cellular differences, as the papillary dermis is more dense in cell compared to the reticular one, and as well fibroblast identity between the papillary and the reticular dermis differs. However, what is not as well known is that there are differences in the vascularization between these two compartments. Skin microvasculature is very essential for skin homeostasis. This includes thermoregulation, immune cell trafficking, oxygen and nutrient supply. It has a very specific architecture that you have here represented. We have a very dense subpapillary plexus that then goes under many capillaries in the papillary dermis that connects a subcutaneous plexus via a network of arterioles and venules. As a result, the papillary dermis is much more vascularized than the reticular ones, and the nature and the morphology of the vessels of these two compartments differ. However, to this day, there is no clear explanation for this difference in vascularization. This is why the aim of this project is to understand what factors can explain the difference in vascularization between the papillary and the reticular dermis. Can papillary and reticular fibroblasts themselves generate a microenvironment that would regulate cutaneous blood morphogenesis? Or are these differences due to a skin-native microenvironment regulation, or even can they both mechanisms explain these differences? To answer these questions, we asked ourselves other questions, including, can papillary and reticular fibroblasts on their own generate a microenvironment in vitro that can sustain the formation of vasculature with the same characteristics that we see in native skin? To answer all of these questions, we decided to carry out a differential analysis of papillary and reticular fibroblasts. We first took a look at their angiogenic properties by using two angiogenesis models. The first one looking at the paracrine secreted factors for both types of fibroblasts in 3D, and the second one looking at the effect of the fibroblast microenvironment itself on capillary formation in a co-culture angiogenesis model. In parallel, we carried out a differential analysis of gene expression and took a look at the angiogenic profile and matrisome of both papillary and reticular in vitro microenvironments. What we know about papillary and reticular fibroblasts is that there are distinct skin fibroblast populations. It was demonstrated using a lineage experiment that they are coming from common fibroblast progenitors that then specified during development. And various in vitro and in vivo studies demonstrated that they play different roles in skin physiology, including aging, wound healing, epidermis differentiation, and cancer. In this study, we used the papillary and reticular fibroblasts isolated from abdominal or breast surgery from 3 healthy female patients aged between 20 and 50 years old. What we did is that we mechanically separated the reticular from the papillary compartments, and we discarded the epidermis. And then after all, we put in culture the explants from the papillary and the reticular compartments. We then defined the papillary and reticular cell cultures based on the morphology and the proliferation rate of the different cell cultures. And so we defined the papillary fibroblast cell culture as a culture with elongated cell, as you can see here, with a high proliferation rate. And on the contrary, we characterized the reticular cellular culture based on the presence of a lot of spread stellar cells with low proliferation rates. In this study, we used both 3 pairs of fibroblasts that we cultivate in over confluent culture, and we stimulated the deposition of extracellular matrix, and I will refer to this culture as cell sheets in this presentation. So, using these cell cultures, we took a look at the angiogenic properties of these fibroblasts using these 2 models. The first model allows us to study the contribution of fibroblasts' secreted factors. It's a 3D angiogenesis model. We are in a fibrid gel, and we use fibroblast conditioned media. And the formation of capillaries in this model is similar to spreading angiogenesis. In the second model, we use a model where we study the contribution of the fibroblast microenvironment themselves. So we let the fibroblasts do their own extracellular matrix, and then we see how it affects capillary formation. And this model is mainly recapitulating characteristics of vasculogenesis. Looking at the first model, so we first wanted to take a look and to see if papillary and reticular fibroblasts secrete distinct factors that could differentially regulate capillary formation in 3D. To do so, we use dextran beads that are coated with gelatin, and we cover them with primary endothelial cells from umbilical cords called UVEC. We then have this nice covered bead that we include in a fibrin gel in the presence or absence of vascular endothelial growth factor. We then add on top of this hydrogel papillary or reticular conditioned medium. And after 4 days, as you can see in this 3D representation, the endothelial cells sprout from the bead and they form in 3D capillaries. This model recapitulates sprouting angiogenesis, the main characteristics, including tip cell and stalk cell specification, the deposition of a basement membrane that you can see here by deposition of collagen type 4, and the presence of a lumen that you can see here on this image of scanning electron microscopy. So you have here the bead, here the capillaries, and the lumen that you distinguish inside. So using this model, what we observed is that the papillary fibroblasts are able to secrete factors that could sustain the formation of a lot of branched capillaries independent of VEGF stimulation. That means that papillary fibroblasts have a really high angiogenic potential. On the contrary, the reticular fibroblasts couldn't secreted factors, had a low angiogenic potential, but were still able to sustain the formation of capillaries in response to VEGF. Looking at the involvement of the fibroblast microenvironment now. So in this co-culture angiogenesis model, what we do is that I take the papillary and reticular fibroblasts that I cultivate as cell sheets, as I mentioned before, and they have the time to deposit a nice extracellular matrix that you can see here on this second harmonic generation image that I did with 2-photon microscopy. So then I sit on top of these layers UVECs, and I allow them to grow and to organize into capillaries. And after a week, we can observe a capillary formation using the endothelial marker CD31. You can see that we have capillary formation on top of fibroblast layers, and in this model we have as well capillaries that are mature, as indicated by deposition of type 4 collagen. I then quantified the vascular network that was done using this model, using a macro that was developed at the laboratory. So to do so, we image the whole cell culture well, and then we choose a region of interest. We then generate a binary mask from which we deduce the percentage of area covered by vessel that I will refer to as vascular area. We then generate an image map from which we can calculate the mean diameter of each vessel. And then from all of this, we generate a skeleton, and by undergoing a skeleton analysis, we can look at vessel length and number of branch points. Using this model, so in the absence of VEGF, there is no difference in capillary formation between papillary and reticular fibroblasts. But you can still notice that both types of fibroblasts in this model can induce the formation of capillaries. What was interesting, though, is that in these conditions, the microenvironment generated by reticular fibroblasts is able to induce the formation of vessels of larger diameter, as verified by quantification. When stimulating with VEGF, we could clearly see that the papillary microenvironment was able to induce the formation of a much more denser vascular network with a lot more branch points, as verified by quantification as well, because we have a higher vascular area, number of branches much more important, and a vessel length as well much more important. So by using these two angiogenesis models, we first demonstrated that papillary fibroblasts have a higher angiogenic potential compared to the reticular ones, and that both papillary and reticular microenvironments in vitro can induce the formation of vessels that reproduce main characteristics of blood morphogenesis observed in vivo. So going further, we asked ourselves, what factors can explain these differences? And to have a more whole approach, what I decided to do is to do a differential analysis of gene expression. So past studies, there are a lot of studies already published on transcriptomy studies about papillary and reticular fibroblast gene expression. And most of them, they demonstrated that papillary and reticular fibroblasts, they differentially express different family of genes involved with different function. And they also identified identity markers, including extracellular matrix components. And recently, there was also a demonstration that there is a switch of identity related to aging. However, these studies were done on non-confluent cells at early passage, and most of them used a microarray approach. This is why, to answer our questions, we decided to culture the papillary and reticular fibroblasts as cell sheets, and then to, as in angiogenesis models, and then to perform an RNA sequencing using the Illumina technology synthesis, sequencing by synthesis, in order to increase the depth of our analysis. So the first thing that we wanted to do is to make sure that in this conditional culture, we still have two different populations of cells that keep their identity. To verify that, we did a principal component analysis, and we observed that the samples segregate into main groups corresponding to papillary and reticular fibroblasts. We then defined the differentially expressed genes as the genes having a ratio superior, fold change superior to 1.5, with a p value that was significant. And we identified 102 differentially expressed papillary genes and 1,244 reticular genes, with more than 50,000 that stayed equally regulated. And among these differentially regulated genes, we found markers that was previously described, such as podoplanine for papillary fibroblast, collagen 7, and for reticular fibroblast, collagen type 11, and FGF7. So we had two different cell sheets with specific identity. Then we wanted to take a look at what biological process that could distinguish these two cell sheets. And to do so, I performed the bioinformatic analysis, and I gathered all the differentially regulated genes that are entered in a David bioinformatic resource. And then I extracted all the Go terms that were the mainly enriched in this condition of culture. And what I observed is that you have here the 20 most enriched Go terms. It's that among the 5 most enriched ones, we found 2 related to extracellular matrix and matrix cells adhesion that are here in green, and also 2 that are related to angiogenesis and its regulation. So it means that in this condition of culture, we have papillary and reticular fibroblasts that are distinguished by angiogenesis and microenvironments. So then we took a deeper look at it. First, taking a look at their angiogenic profile. To do so, I listed all the genes that were differentially regulated for each subtype of fibroblast, and I compared it to the list of the vascular development Go term. Then realized an expression heat map and carried out a hierarchical clustering. You have here a representation of this study. And what I observed is that spontaneously, the samples clustered in 2 main groups corresponding to the papillary and reticular fibroblast samples, indicating that we have a specific signature for angiogenic properties. Then to go further, we asked ourselves, okay, we have difference in genes, but are these genes pro or anti-angiogenic factors? So then I extracted from these genes that I referred as effector of angiogenesis. So effector of angiogenesis, I define them as genes coding for proteins that are able to interact directly with endothelial cells, including secreted factors, ECM components, as well as membrane and core proteins. And to this day, I found almost only pro-androgenic effector gene expressed in the papillary fibroblast, while I observed a more subtle balance between pro-anti-androgenic genes and context-dependent genes. So these differences could explain partially the difference that I observed in the androgenesis model in vitro. And interestingly, I found expression of smooth muscle cell differentiation markers within the reticular fibroblasts, markers that are expressed in some vascular smooth muscle cell associated cells, and that are really important to understand, that could explain as well differential interaction between the endothelial cells and the reticular fibroblasts. To partially verify this with protein, I did an antibody array using conditioned media for papillary and reticular fibroblasts. It's a commercial antibody array with 53 target proteins, and I confirmed the differential expression of some proteins analyzed in RNA sequencing, including collagen type 18 and placental growth factor for papillary fibroblasts, hepatocyte growth factor and chemokine ligand type 2 for reticular fibroblasts. And interestingly, I also observed differential secretion of other non-identified targets, such as VEGF for papillary fibroblasts and interleukin type 8 for reticular fibroblasts. So now, we know that with this bioinformatic analysis, we have different expression of pro, anti, and context-dependence markers between the two fibroblasts, including angiopoietin type 1, anti-androgenic regulators in reticular fibroblasts, such as thrombospondin type 1, BMP4, and context-dependence, such as NHPCL, like angiopoietin-like 4 protein. As I mentioned, the matrix is as well another differentially regulated process. So then we took a look at this process using the matrisome. So the matrisome is a list of genes that lists all the genes that are involved in the structural composition of the matrix and its regulation. And it's available online and published by Naba et al. And it's subdivided in different categories, such as the secreted factors, regulators, ECM affiliated. And the matrisome itself can be subdivided in between glycoproteins, collagens, and proteoglycans. So in a previous study carried out by my teams, there was a matrisome that was characterized for the freshly isolated papillary and reticular fibroblasts. And in this study, we identified the markers of ECM from these two types of fibroblasts, and we also observed that the reticular fibroblasts express more genes related to the matrisome, and especially structural components of this matrix. So taking this as a reference, we then took a look at the matrisome of the cell sheets that we generated here, and we had an increase by 4 times of the number of matrisome-associated genes. And interestingly, this difference was not equal between papillary and reticular fibroblasts. We find 5 times more genes in the reticular fibroblasts an increase, and only 3 times in papillary fibroblasts. And this, we found more ECM-associated genes. And looking at the structural composition, so the core matrisome, we found the same trend, and with more glycoproteins. So now, the same as for the androgenic profile, I took a look at I made the heat map expression of all of these genes, and I'm presenting you for each subcategories the results that I got. So using hierarchical clustering, what we could observe is that there was a spontaneous, as well, gathering of the samples according to papillary and reticular identity, meaning that in this condition of culture, we have papillary and reticular microenvironments that have specific matrisome signature. We then found as well previous markers that were defined, especially in the P1 study that I here referred in purple. Interestingly, we find twice more non-fibrillar collagens expressed in the papillary fibroblasts. And we found as well 6 times more proteoglycan that were differentially expressed in the reticular fibroblasts. And we enriched the number of genes associated to the elastic fiber assembly in the reticular fibroblasts that are here highlighted in red. We did the same, and I'm still doing the same for the matrisome-associated genes. And to this day, we have very interesting lead. And I noticed the presence of genes coding for protease inhibition of in the reticular fibroblast, and notably as well identified actors of immunity in the ECM-affiliated genes. So to summarize what we learned with this study, so using two models of angiogenesis, we demonstrated that papillary and reticular fibroblasts have different angiogenic properties. This is due to a higher angiogenic potential in the papillary compared to the reticular one. Plus, by their own in vitro, generating their own microenvironment, they can reproduce some of the features that are characteristic to cutaneous blood morphogenesis. To this day, using RNA sequencing, we here propose that these differences are due to a difference in expression of gene involved in the pro- or anti-angiogenic regulation of angiogenesis, with more genes involved with pro-angiogenic in the papillary versus anti-context-dependent in reticular fibroblasts. We also identified markers of smooth muscle cells that could explain that could be responsible for different interaction between the reticular fibroblasts and the endothelial cells. And we demonstrated that they have a specific metrisome. So what's going next is to understand what functional links can we make between the microenvironments and the regulation of angiogenesis in the papillary and reticular microenvironments. And this is the first step to understand what is the regulation of the formation of vessels within the skin, and it's very important for tissue engineering perspective and even for therapeutic perspective. So I would like to thank you very much for your attention and to thank everybody that contributed to this work at the Collège de France, at the IGFL, and at CILAB. Especially thank for Pauline Joncourt, who did all the biostatistic work. Thank you to the engineers of the platform sequencing at the IGFL de Lyon, and thank you very much for the past and present engineers of the core facility at Paris. And I'll be happy to answer questions.
Thank you. Some few questions, because I have. So just, you mentioned that therapeutic application.
Yes.
Which kind?
So it's future application. What we can think about is what kind of fibroblast for what kind of graft. For example, as we see that the papillary fibroblasts have a much higher angiogenic potential, what we can think of is for a chronic wound, we can preferably do graft with papillary fibroblasts compared to the reticular one. And I think it's also important to emphasize that as the reticular fibroblasts by themselves can also do blood morphogenesis, perhaps in few years we will be able to find applications for reticular fibroblasts themselves in other contexts. To this day there is none, but I think this is going to come as well.
Could we consider that one of the population controls the other one?
Yeah. So this is really interesting, and definitely this is something that we have to look at. In the previous study, in transcriptomic, it was demonstrated that there could be some crosstalk looking at the regulation of some signaling pathway. And I would definitely say that it's more likely that there is a communication between the two subtypes to maintain the blood structure. Particularly because in the skin, it's true that in adult skin, there is no angiogenesis except in disease or except in wound healing. So I would say there is kind of a status quo between the two compartments.
And the last point, after that I stopped you. So, and what about neuromediators? So you don't mention CGRP, NPY in the control of all this angiogenesis, because you don't find anything in your global screen? Or?
So I didn't find differential expression in this screen. And it's important to, I would like to emphasize as well that I only took a look at the differential expression. But it might be that right now I can answer because I don't have in mind the list. But I would not be surprised if there is an equal regulation of these factors between the two types of fibroblasts.
Yes, I think so.
I have a question about your RNA-seq analysis. Did you analyze the data at the gene level or at the transcript level?
Wait, sorry, at the gene level?
Gene level or at the transcript level? So do you think it could be of interest to analyze the data at the transcript level and not only at the gene level?
Do you mean the level of transcripts?
Yeah.
The number of reads that I will find for each?
Yeah, you have the reads, but you can summarize all the reads for one gene, or you can identify the different transcript, potentially differentially splice or variant of transcript for each gene. So I would like to know if you could be of interest of doing this sort of analysis.
So here is a level expression of genes, but not spliced variants. This is the question, right? So I didn't do it here. Yeah,
so it depends the way of sequencing, if it is per hand or single hands. But if it is per hand sequencing, you can have the gene variants in your data.
I don't have the gene variants here.
Yeah. Okay, thank you.
But it's definitely something that we have to look at. That would be interesting.
So just a comment before
yes.
I think that so we have a session this morning on microbiome control, and so people work on this kind of approach to understand neovascularization of the skin, but nobody's use any kind of strain extract or strain mediator coming from the local commensal population to try to know if yes or no there is an increase of vascularization that this kind of strain can provide something to the skin, even in deeper, because so Rich Gallo demonstrated a few years ago that we could find some bacterial strain until the basal layer. I think that maybe you could have a look on this field. So.
And it's particularly interesting because we found some immunity markers that are well known in papillary fibroblasts, right? So, including antigen recognizing molecules. That definitely would be really interesting to look at.
Maybe you could exchange with the guy this morning, coming from Lund.
Yes.
Okay, thank you.
Thank you very much.
The last one.